have both a PhD and a bachelor's degree in biochemistry, which you obtained at the University of Calgary in Canada. And you're going to be talking to us today about mTOR at the intersection of nutrition and energy expenditure and obesity. With that, I'll let you go.
So, is my voice okay carrying back there? It's okay, so I don't need the mic. Perfect. Don't have to worry about me turning and scratching the mic with my collar every time I turn my face. Right, so thanks Peter and Karen for inviting me to come down here. It's nice to be back. I was just saying, the nice cool air is nice. In Memphis, it's hot and muggy, and being from Canada, I'm probably never going to adapt to the hot and muggy of Memphis. So it's nice to be out on such a nice day. And good luck with the snow next week, if you guys get it. So what I want to talk about a little bit is some of the studies we've been doing in my lab. I've been at the University of Tennessee for a little over 2.5 years now. And some of the studies we've been looking at about nutrient sensing and how that relates to energy homeostasis and metabolism. So I think most people can appreciate the balance that I'm showing here, that in order to maintain neutral energy balance, you have to have energy intake roughly be equal to energy expenditure. And when you're in cases where your energy intake is a little too high, because, for example, you move to Memphis and you discover like barbecue pork ribs, your energy intake goes up a little much, and then you get a faculty job and suddenly you have less time to jog, and your energy expenditure doesn't quite match. And so that's what we call positive energy balance, and that generally results in weight gain. And then when we want people to lose weight, we generally tell them to reverse it. So we tell them to eat less and to exercise more. And so that's key to people who are obese and maintaining their energy balance. And like I said, what we usually tell people is to focus on their eating and their physical activity. And those are two parts of the intake-expenditure balance that are really, really important. But what I want to talk to is two parts that are actually a little harder to control. So these are sort of the metabolic parts, and that's how energy is actually stored. So when you eat food, what you actually do with the food you eat, and then what your basal metabolic rate is. So what is your energy expenditure outside of the actual exercise that you're doing? What is your basal metabolic rate? And how can we control those two things, and how can those two things predispose somebody who's maybe not eating way too much or not super inactive to getting obese? And so the way we conceptualize this, the way we think about this, like Peter said, I'm a biochemist, so I come from a very molecule-up background. And so in my PhD, we did a lot of cell stuff, a lot of enzyme stuff. And then as I did my postdoc with Alan, we moved kind of into cell culture experiments, trying to understand how things work within a cell. And that's still kind of how I think about things, except that we're trying to expand to understand how those things work in an integrated way. So if you think about how the organisms have to establish a balance between nutrient levels and nutrient responses, the first step is you have to be able to sense the nutrient status. And this happens in every cell in the body. Every eukaryotic cell has a way to modify and sense how much nutrients it has, how much is around, and what it should do. So that's the first thing. And then the second thing is be able to respond to that nutrient level. So if you think of it from a yeast perspective, you can think a yeast has to be able to figure out, is there enough nutrients around? Should I divide? Should I not divide? Should I get bigger? Should I store things for later? And it needs to be able to think about that in a coordinated way. Now, within a yeast cell, it's pretty easy, because within a yeast cell, it just has to think about itself. It just has to survive, it has to divide. It doesn't have to think about how to coordinate its metabolism with, say, the yeast cell over there, because he's on his own. He's over there. Unfortunately, in the human body, they have to talk to each other. So what your muscle does can't be completely dislinked from what your brain does, what your fat does, what your liver does, what your pancreas does. And so the coordination between how nutrient sensing occurs and how that regulates overall metabolism is something that we think about too. So that's kind of how we conceptualize the intersection between nutrients and metabolism, is we sort of think from inside the cell out to the rest of the body. Now, that's not to discount the fact that nutrient status sort of in the serum is regulated too. We think of it just as the other way. We think of it, what does the cell think is happening, and then how does that affect what the cell does, and then consequently what the rest of the organism does. So I'm going to talk to you briefly about how different cell types could respond to nutrient status. And to understand what I'm going to talk about, there's this nutrient sensing kinase called mTORC1. So it's the mechanistic target of rapamycin complex 1. It's called the target of rapamycin because it is the target for the drug over there that is called rapamycin. Rapamycin is called rapamycin because it was from Rapa Nui, which is from Easter Island. So that's why it's called mTORC1. So most of that abbreviation is totally irrelevant for what it actually does, but that's how it was named and that's what it does. And that's going to be the focus of pretty much everything else I'm going to talk about for the rest of the day. So what does mTOR do? So it's a really interesting nutrient sensing node. And what it does is it can sense the levels of both extracellular signals like growth factors and insulin, and then also intracellular signals like ATP or glucose and amino acids. And it can integrate all those signals together in that if you have lots of insulin and lots of amino acids and lots of glucose, you get more mTOR signaling. But if you decrease any of them in an independent way, they can all modify it. So what I mean is that even in a state of lots of insulin but no amino acids, mTOR signaling will be down. So it's this nice intersection point by which a lot of different nutrients kind of group together to integrate metabolism. Now, mTORC1 is really old. It goes all the way back to yeast. So basically, anything that it did initially happened in yeast. And so when we think about how mTOR functions, quite often I think of it as there was an initial nutrient sensing module all the way back in yeast. And then evolution has just kind of added extra bits and pieces onto it to make it more complicated. So that now in humans, it responds to a bunch of other stuff. But at least in yeast, it had to know what were the internal amino acid levels, what was the internal ATP levels, and then that's how it's going to integrate that signal and respond. And so its response in general is, I think of it as to alleviate nutrient excess. So if a yeast cell has lots of mTOR, it's because it's sensing there's lots of nutrients and it has to do something with those nutrients. If nutrients are not around, there's not very much mTOR activity, and therefore it has to do kind of the opposite things. And there's a lot of things that it does. I'm listing a few on the side here. It regulates lipid and glycogen storage. It regulates autophagy, which is kind of a way to break down large complexes in the cell. It regulates making mitochondria, and it regulates extracellular events like insulin resistance. So what I'm going to talk about now is sort of how different cells are going to have different responses when you think about what mTOR does. So again, I think of mTOR as there's a level of nutrient excess, and then there's sort of mTOR-dependent nutrient responses. And in general, those things are going to either store nutrients or burn nutrients. But the ultimate goal is to alleviate that nutrient excess or alleviate that nutrient stress. The converse is also true. So if you don't have enough nutrients, you're going to stop the storing and the burning of nutrients, and so therefore you have more nutrients available to do the important things. Now the reason I'm kind of belaboring this point a little bit is that I didn't really appreciate this until actually we started making various knockout mice that I'm going to talk about. But if you think about it from the concept of an adipose tissue cell, so a fat cell, the way it's going to respond, its mTOR-dependent cellular response, is going to be to store fat. That's what it's going to do. A fat cell is a machine that is finely tuned to make fat. And so the way its mTOR is going to work is going to be to store more lipid. But if you think of a muscle cell, its main function is not to make fat. Its function is to make ATP and use it for energy. And mTOR in muscle, as I'm going to tell you in the next few slides, its role is going to be to burn as fuel. So whereas in both cases, it's trying to accomplish this, what it does in individual tissues is really different. Now that may seem obvious now. Maybe it was obvious to you guys all a few years ago, but not obvious to me until we started working on this, because I was thinking a little bit too much of the cell out. So again, the way we try to think of it is there's an integrated node, but the way that node of mTOR signaling does its job is context dependent. And that's what we've been trying to understand, how that matters in metabolism. You could also think that in a pancreas, you're going to do something different than a muscle. So in the pancreas, mTOR regulates insulin secretion. And in the brain, it alters your behavior. It alters your food-seeking behavior. So I'm not going to talk about the last two. I'm just going to talk about the first two. We're going to talk about what mTOR does in adipose tissue, and we're going to talk about what mTOR does in muscle tissue. And we're going to talk about both of them, or me. I am going to talk about how these things affect sort of overall metabolism systemically. All right, so we're going to start with muscle. So the way I want to sort of contextualize what I'm going to tell you about muscle metabolism is what you could think of as what are the normal responses to overfeeding. So in Alan Saltil's lab when I was a postdoc, he spent a lot of time thinking about what insulin does. And you could think of insulin, or the sort of anabolic roles of mTOR, as anabolic responses. So you have extra nutrients in your blood, extra glucose, extra lipids, and you got to put those away. You got to put them somewhere. So you do glucose uptake, you stop making new glucose in the liver, you store lipids and glycogen away in other tissues, you make synthesis. Kind of at a longer term, you might make more fat cells, you might even make more muscle cells. So all of those you can think of as like anabolic responses. These are things that are going to store things away. And in general, the anabolic responses, again, feed back to restore nutrient homeostasis. If this doesn't work, in the case of obesity-related type 2 diabetes, the anabolic responses aren't as effective. So you can't clear these things out of your body. Your blood glucose ends up going really high. On the other hand, there's also a series of catabolic responses. And this is, I think, something that I didn't appreciate nearly as much, that there's signals when you have elevated nutrients that are going to increase your metabolic rate as another way of retaining nutrient homeostasis. Now you can think in the case of obesity, you actually don't necessarily want to be activating too much of the anabolic responses, making more fat storage. Maybe it helps get the glucose out of your blood, which helps with the type 2 diabetes, but it's going to exacerbate the obesity problems. It's actually a little more useful if we can think about how can we stimulate the catabolic responses, because that's a better way of getting rid of the nutrients, is burning it off as heat. So here's an example of what I mean by the catabolic responses. This is a paper from 1988. And so what they did was they took humans and they looked at their energy intake, which is this bar here, and their energy expenditure, which is this hatched area here. And then they did an overfeeding experiment where they gave them every day, they tried to give them more and more carbohydrates, and then they wanted to see what happened to their energy expenditure. So you can see, starting here, every day they gave them more and more carbohydrates, and every day their energy expenditure kept increasing and increasing as they were given more of these carbohydrates. Now they were still in net positive energy balance. You can see the amount of energy they were taking in was still high, so this wasn't enough to compensate for it, but there was this increase in metabolic rate that was in response to this nutrient excess. So if you think about, well, what happened to that stuff, if you look at what they looked at, what happens to the carbohydrates they're giving them, you see initially there's a very rapid, pronounced increase in the rate at which these carbohydrates are oxidized, the way at which they're burned as fuel. And that happens very quickly, very early, and it goes from here to here. So there's a very big, robust change. Sort of at the same time, maybe a tiny bit later, there's an increase in storage of glycogen, and then a little bit later than that, there's an increase in net de novo lipogenesis. So the first thing in this experiment the body tried to do was to basically burn that fuel, to get rid of those carbohydrates. And then once eventually it got to this point, it sort of gave up and said, all right, we'll put it away as fat because we can't burn carbohydrates anymore. So this is another experiment from Jules Hirsch, and it's sort of showing the exact same thing. So they took people, they made them gain 10% of their body weight, and then they looked at their rate of energy expenditure by indirect calorimetry. And as you can see over there, with a 10% increase in weight, they got a big increase in their metabolic rate, their rate of energy expenditure. When they returned back to their normal weight, it went back to normal. And if they made people lose weight, either baseline or after returning to their initial weight, their metabolic rate decreased. So what I'm trying to get across to you is this idea that the body is responding to your nutrient levels, your nutrient intake levels, and modifying its metabolic rate accordingly to try to have a catabolic response. And so this is really important if you think of it in the context of somebody trying to lose weight. So if you start with an obese person, and then they lose weight, like I showed you on the previous slide, what's going to end up happening is your basal metabolic rate is now going to be lower. You've lost weight. Your body sort of thought, like, hey, wait, I thought there were more nutrients around. I better slow things down. Now, along with sort of residual increased feelings of hunger, you're going to be much more likely to bounce back up. You're burning less fuel. You're a little bit hungrier, so it's more likely that you're going to revert back to being a more obese person. And that's when we think about the set point problem. And when we want people to lose weight, but the set point never changes that much. And people have done these experiments, these indirect calorimetry experiments, where they force people to lose weight, and they've seen the decrease in metabolic rate. And then going out 8 years past the weight loss, their metabolic rate still hasn't bounced back up to what you would expect from a lean person. So this is like sort of a chronic thing, and the mechanisms that sort of link the nutrients to the metabolic rate are not that well understood. And so we think that this is a pretty important problem. So the way we think about it again is we're trying to understand this from a cell signaling perspective. We're trying to understand what are the mechanisms by which we can modify energy expenditure, hopefully in a way that we can generate a negative energy balance to help somebody lose weight or to keep weight off. So before I get to mTOR, there was one more hint that kind of led us down this direction. So this is a similar experiment to the one I showed you where there was the weight loss. So in a normal case, they made the people lose weight, and then they put them either on a high carbohydrate diet or a low carbohydrate diet, which ended up being a high fat, high protein diet. And if you look at the energy expenditure, so this is after they both lost weight, and they're given equal calories of these two things, you see that the high fat, high protein group actually had less of a reduction in energy expenditure. So their energy expenditure stayed a little bit higher, a little bit closer to what it was before they lost the weight. If you think about it from an overfeeding perspective, this is from a George Bray paper, again, they overfed them with the same number of calories, and then they looked at what was their increase in metabolic rate. So in the case of the low protein, or you can think of high carbohydrate group, they had a small increase in energy expenditure, but if you look at the high protein group, they had a much larger increase in energy expenditure. And this has been borne out in several other studies since. So what this suggested to us is that it wasn't the amount of calories per se that was modifying the energy expenditure, it was the source of the calories, the macronutrient composition that was somehow transmitting the signal to the rate of energy expenditure in the intact person. And so we tried to repeat this. We tried to do a similar experiment in mice. So we took mice and we gave them a matched either control diet, which had 10% of its calories came from protein, or a high protein diet, which was 45% of its calories from protein. And then we looked at what happened to the mice just on that diet. So what I'm showing you here is the amount of fat based on MRI imaging. And you can see the control diet mice gained quite a bit of fat, and the high protein diet mice were resistant to the sort of increase in fat mass. They had smaller adipose tissue if we dissected it, and they had slightly larger muscles when we dissected those. They also had higher energy expenditure. So this sort of suggested to us that there might be some link between protein specifically as a macronutrient and these changes in energy expenditure and sort of anti-weight gaining effects. And so, again, the reason we're interested in this is because one of the most strong signals to activate mTOR is amino acids. So amino acids, as the breakdown products of a high-protein diet, are increased. And so we wanted to ask, well, maybe that's the way by which amino acids respond to a nutrient excess, is by promoting energy expenditure through mTOR. And so there's reason to believe mTOR is activated by amino acids. This has been shown by many different groups. This is just our way of showing it in cultured muscle cells, where you can see if we look at the phosphorylation of a downstream target, if we remove amino acids, mTOR signaling goes down by Western blot. If we add amino acids back, mTOR signaling goes back up. So it's a very acute responder. This is about half an hour of the added amino acids back, a quick acute response to that, to amino acid levels. If we take now the tissues, the muscle tissues from those mice that I talked about, and we looked at the mTOR activity by the phosphorylation of S6K, you can see the high protein diet mice, even after we've taken away the food for overnight, still have higher residual mTOR activity, and it's increased almost to the level of the difference between the fed and the fasted state. So that's what the quantification there says. So what these two pieces of data sort of suggested, that in muscle cells, both in cultured cells or in vivo, protein or amino acids could modify mTOR signaling sort of in a positive way. So the next we decided we needed an assay for this calorie-induced change in energy expenditure. So this is an indirect calorimetry experiment with some mice. So mice are, during the day, because mice are nocturnal, are sleeping, so their energy expenditure is quite low. If we switch them, or if they move to the night cycle where they're more active and they're running around, their energy expenditure increases, and then they go back to sleep and their energy expenditure decreases. If we switch them from chow, which is a sort of nutrient not dense food, to a high-fat diet, which is a calorie-dense food, you can see their energy expenditure jumps up within that first day. So there's an immediate response to the high-fat diet to increase their rate of energy expenditure as measured by this calorimetry. Now you may think, well, maybe what they're doing in response to this increase in energy content is they're running around more and they're burning that fuel. They've decided that, oh, that was a good ice cream sandwich. I'm going to go hop on the mouse treadmill and jog. And that's not the case. So if you look at their physical movement, so this is measured simultaneously to their metabolic rate, their physical movement doesn't really change that much in the context of this experiment. So this seemed like a nice paradigm by which we could see sort of a quick pro-energetic response in response to a diet in the absence of changes in physical activity. If we think about what happens to mTOR now in the context of a high-fat diet, if you look at the phosphorylation of its substrate, this is now in liver, you can see that there's an increase in mTOR activity after a high-fat diet as well. So you can see this is darker than this, which is the phosphorylation of S6K. So mTOR is also activated by this high-fat diet. So then we wanted to ask the question, well, what would happen if we block mTOR activity in this assay that we're looking at? So this is a bit of a complicated slide, or a bit of a complicated picture. But this, again, is some indirect calorimetry where it's like night cycle, night cycle, night cycle, night cycle. And then after 4 nights, we switched them to the high-fat diet, which is right here, and then we measured what their calorimetry was, their energy expenditure was after the high-fat diet. Now again, you can see there's this increase. Their energy expenditure increases in response to the diet. But if we randomize the animals into either a vehicle-treated group or a group that we treated with rapamycin, we did that right here. And then if you can, it's easier to see on the bar graph there. If you look at their increase in energy expenditure, the rapamycin-treated group doesn't have this increase. So again, remember, rapamycin is this drug that specifically inhibits the mTOR complex. Now, it inhibits it everywhere. It's going to inhibit it pretty much everywhere in the mouse body. This doesn't tell us anything specifically about muscle. It just says mTOR somewhere, somehow, is involved in this increase in energy expenditure. And then again, there was no change associated with physical activity. So this is purely a change in their metabolic rate. So we wanted to ask, what would happen if we looked at the effects of mTOR directly in muscle to try to figure out what was happening in energy expenditure. So again, what I've tried to tell you is that overfeeding, and specifically overfeeding with protein-rich diets, can cause some activation of mTOR in the muscle. And I've also said, we think that maybe what's happening is that there's an activation of mTOR muscle, and that is increasing the oxidation rate of fuel. And that's increasing the metabolic rate, that's being burned off as heat, and that's how the metabolic rate is increased. Now, I'm not ruling out that there's something else. So like that arrow, you can imagine there could be anything else, everything else, sort of all the other hypotheses. And people have known sort of what we've been suggesting about the overfeeding of protein and the activation of mTOR and the increased oxidation rate from studies in people for quite a while. But we wanted to know sort of mechanistically, well, is this sufficient for this increase? Is this mTOR effect really important, or is that lower arrow also really important? So what we were able to do is we can have a mouse system where we activate mTOR in muscles specifically and then ask what happens. So now we're not giving them a high-protein diet. We're not putting them on a high-fat diet. We don't have to worry about all of those confounders. We don't have to worry about any other confounding effects of the high-fat diet. So that's what our model was to set up a system where mTOR was sort of constitutively activated in muscle and then ask, well, what happens to those mice metabolically? And so the way we can do this is that mTOR has this really important negative regulator. It's called TSC1 or TSC2. There's two genes. They're both essential. And it acts as a brake on mTOR activity. So when TSC is there, it keeps mTOR activity nice and low. If you delete TSC1, which we can do in mice with tissue-specific knockouts, you take away that brake, and now mTOR activity goes way up. Right? So this is the way that we accomplish the activation of mTOR. So there may be other TSC-dependent effects in the cell that are mTOR-independent. That's possible. There isn't really that many that are described that we think are relevant, but this is the way, because TSC is so specific, we've activated mTOR in the muscle. And sort of just to show you that it works, this is some Western blots where you can see in the knockouts we've deleted TSC1, it's gone, and the phosphorylation of S6K and S6 are both increased. And this is in fasted muscles where mTOR would not normally be active. So this is our way of activating the muscle, making the muscle think that it's in a condition of nutrient excess, even though it's actually not in a condition of nutrient excess. Does that make sense? Cool. All right, so then we looked at the energy expenditure of these mice. And this was actually done here. So this is how long this study has taken. I started this experiment here. Nathan did it at the MNORC. And then I kind of ignored this VO2 data for 2 years because I'd forgotten that we had done this experiment. And then when we were trying to put together a few months ago what we wanted to say about the mice, I was like, I kind of vaguely remember doing this calorimetry experiment like 3 years ago in Allen's lab. And it, at the time, didn't make any sense. But if we look at the calorimetry from these animals, again, you can see their VO2 in the muscle-specific knockouts is already basally increased. So the red line is much higher than the black line. And again, this isn't because they're more physically active. They're not running around burning this fuel. They're just burning that oxygen just at a basal level. So what this suggested to us is that activating mTOR in the muscle, that alone was sufficient to increase the metabolic rate. Now this doesn't rule out that lower arrow I was showing you, where it could be also due to something else. To do that, we need to inhibit mTOR in the muscle, and we're still working on that. That's actually taken a little bit longer than we had expected. But this together suggested that what we're seeing is just activation of mTOR in muscle is able to increase the oxidation rate sort of at a whole body level, presumably by increasing the oxidation in the muscle, because that's the tissue we're affecting. So then we wanted to ask, well, what does that do to the actual animal, right? The reason we're not doing this in cells, only in cells, is because we want to know, well, how does this matter overall? If this doesn't have any change on glucose regulation or response to high-fat diet or body composition, then we kind of don't care. Now we've just found this random phenomena that doesn't really make any difference and doesn't matter. So fortunately, because we had done these knockouts in mice, we could look at what else was happening in the rest of the animal. So if we look at the growth of these animals, and again, I'm just showing you the fat mass here, up until about 70 days, the mice are about the same amount of fat. They're not that different. These lines make it seem different because of the best fit. But these points are basically totally overlapping. Now the wild-type mice, so these are the mice that have normal mTOR activity, they continue to accumulate fat throughout their lifespan. Basically after 30 weeks, that line kind of keeps continuing. But the knockouts, even though their fat mass was the same sort of through development, at about 70 days, or 70 days and about 10 weeks, they stop accumulating fat mass and it basically stays pretty stable. If you quantify this, this is now done at 20 weeks, this quantification. See there's about a 50% reduction in both fat mass and percent fat mass. And there's no change that we could detect at all in the amount of lean mass. So this is one of those paradoxical things. We made a big effect, or we made an effect in muscle. We expected to see a muscle phenotype, but when we looked at the animals sort of from a composition standpoint, the main effect wasn't in the muscle. The effect we're seeing was in the fat. This is what fat pads look like from mice. So this is epididymal and inguinal fat pad. So an epididymal fat pad is what you would think of as like a visceral fat pad, like belly fat. And an inguinal fat pad is a subcutaneous fat pad. BAT is brown adipose tissue. So that's what they look like from a normal mouse. You see they're white, they're nice and triglyceride rich. And that's what they would look like from a littermate of those mice. You see the fat pad is much smaller, it's much less white, there's much less triglyceride, and it looks much more red. And obviously, like much, much smaller. Just to show you that I'm not just picking a random example, we quantified this, I think, off 25 or 30 mice, and you see there's about a 70 to 80% reduction in fat mass when we look at the dissected fat pads from these mice. So they have much, much, much less fat than their wild-type mice. And again, this was all done at approximately 20 weeks. So again, what we thought was happening was that we're seeing an effect where we know genetically the effect is originating from the muscle. So we've done the controls to show that we're not accidentally knocking it out in the fat tissue, we're not accidentally knocking it out the liver, or the brain, or the pancreas. We're specifically intervening here, but we're seeing a really dramatic effect way over there. So we're affecting sort of fat tissue metabolism and fat tissue content by starting with the muscle. So this is a way that in animals we can sort of separate the effects. If you did just exercise, you would expect systemic changes in all sorts of these different organs. But in our case, we can see, starting in the muscle, an effect on fat. So we have a couple of different hypotheses about how this could happen, sort of functionally. So one hypothesis is that there's a hormone, in a muscle-derived hormone, the myokine, and that is being secreted from the muscle tissue that is then going to the fat, signaling to the fat to tell the fat, well, you should get rid of that triglyceride, you should burn some of that extra triglycerides. So that's one hypothesis. The second hypothesis that we're thinking about is that the muscle is just acting as a sink. So it's acting as a lipid sink. It's got more metabolic activity, and instead of lipids being diverted to fat, they're just going to the muscle and they're just getting burned off as heat. So that's our second hypothesis. Now, we've looked at, there's several myokines that have been described previously that have been suggested to regulate adipose biology from muscle tissue, things like IL-6 and irisin and meteorin-like protein. We've looked at the levels of all of these, both at the RNA level and at the serum level, and for none of these, for all of those, we've not seen any changes in the levels. So at least for the known myokines, we've not seen any differences. So right now we don't have any positive data to suggest that that hypothesis is true, although we can't really rule it out. What we do think is happening is the lipid sink hypothesis. And one of the reasons we think that is we look at some markers of some oxidative fiber types. These are a myosin and a circa isoform that are specific to oxidative fiber types. We see a big increase in expression of both of these markers, and really all of the markers of oxidative muscle tissue. And so muscle tissue, to remind people, is broken down into different types of fibers. There's oxidative muscle fibers, and there's glycolytic fibers. The oxidative ones are really dense in mitochondria, and the glycolytic fibers have much less mitochondria. And this is from a different paper that used a very similar knockout mouse to ours, where they also knocked out TSC in muscle. And what you can see, if you look at the staining for NADH, you can see that both in the TA muscle and in the soleus muscle, there's an increase in the number of oxidative fibers, and those oxidative fibers, the blue fibers, are much bluer than in the wild-type animals. So we think what we're seeing is a switch either towards more oxidative fibers, or the oxidative fibers that are there are more mitochondrially rich, and we're still trying to sort this out, exactly which of those two things, or both, are actually positive. Now, when you have more mitochondria in the muscle, now that makes your mitochondria more able to burn the fuel. So we think that that's the underlying reason why the mitochondria, or why the muscles are acting as a sink to suck the lipids away. Okay. We okay? Does anybody have any questions? All right. Okay, so what I've tried to tell you in this part is overfeeding responses can activate mTOR in muscle. This happens in response to a high-fat diet. It happens in response to a high-protein diet. And that causes this increased oxidation rate. Now, what we think is this is a normal homeostatic feedback as a way to respond to overfeeding. So we think that this is a way that animals that have muscles are able to sense at a muscle level that there's more nutrients available, increase the amount of oxidative fibers or the oxidative capacity of those fibers, and then burn some of that fuel so that the overfeeding responses don't result in excess fat accumulation. All right, so moving on to the other tissue that we've looked at is adipose tissue. So we've basically wanted to ask the exact same questions in adipose tissue. We wanted to ask, well, what happens if we affect mTOR signaling in adipose tissue? Is that going to be any different than if we affect it in muscle tissue. And so the context for this is going to be a little bit different. So I'm going to give you a little bit of a different paradigm for why we think excessive lipid storage in adipose tissue is functionally relevant. So if you think of what happens when you have a meal and you have excess circulating glucose levels, there's a response largely mediated by insulin, where you're going to stop glucose production, you're going to prevent gluconeogenesis, you're going to promote glucose disposal, so glucose uptake, and you're going to promote lipogenesis and glycogenesis. So you're going to try to get that glucose out of the blood by putting it into the liver and the fat and the muscle tissue. And then those things are going to feed back to sort of lower glucose levels. So this is your sort of normal insulin signaling cascade. So if you thought in the context, well, what if there was no insulin resistance? What would happen is as soon as you started that cascade, glucose levels would drop and they would kind of keep dropping and keep dropping. So like with almost every endocrine effect, there's a negative feedback loop to prevent this from happening. And this is what we call insulin resistance. So insulin resistance, as you might expect, just works to kind of do the opposite of what insulin does in each of these cases. So it's going to work to make more or less suppression of glucose production and sort of prevent glucose disposal and nutrient storage. Right? Does that part make sense? All right, so this is what you would expect if you had if you had insulin resistance. And when you have too many nutrients, you become obese, insulin resistance goes up and up and up. So what you might expect is if you have too much insulin resistance, you would see something like that, where you get a big increase in glucose production, you get a decrease in glucose disposal, and you get a decrease in the insulin-mediated effects of nutrient storage. So that all makes sense. That's what you'd expect. The first two things definitely happen. You get aberrant increases in gluconeogenesis. You get impaired glucose disposal. That results in elevated glucose levels, which is why you get type 2 diabetes. So that whole part makes total sense. But what doesn't happen when you're a type 2 diabetic is you don't stop nutrient storage. People don't become type 2 diabetic, at least until they get to the catabolic phase, and then immediately become skinny. They stay obese, and that obesity is maintained, and that's a problem. So we wanted to think about, well, how is nutrient storage still able to happen, even though the effects of insulin are largely attenuated? So some evidence that this is happening. So here is some high-fat diet mice, and in this case, we looked at glycogen, but it's true of triglycerides as well. And if we looked at the levels, this is in liver, after a high-fat diet, you see after a high-fat diet, there's more glycogen storage, there's more lipid storage in the liver after both cases. So these animals are very insulin resistant. So they don't have insulin-stimulated lipogenesis and glycogenesis, but they have higher steady-state levels of glycogen and triglycerides. So you may argue, well, that's probably just because they have more glucose going around, and that's just going in there because you have more substrate. So we repeated this experiment, but what we did is we isolated soleus muscles from an animal that was either on a normal diet or a high-fat diet. So this is a little complicated, so I'm going to take a little bit of time to go through this. If we think about just the high-fat diet animals, so we took soleuses out, now we don't have to worry about whether the circulating glucose levels are different, because they're in a petri dish where the circulating glucose levels are controlled. And we're only, in this case, measuring the anabolic action. So we're measuring radiotracer glucose uptake and incorporation, in this case, to glycogen. It's true for lipid, too. So we're not worrying about whether we're impairing catabolism. We're only measuring whether the anabolic rate is increased. So normally, you have an increase in glycogenesis in response to insulin. You can see the normal chow animals have this nice, robust response to insulin. If you look at the high-fat diet animals, they have effectively no insulin response. So in this case, they're, to the extent we can measure it, totally insulin resistant. But the difference is what we're seeing is at the basal level, there's this big increase in basal glycogenesis and basal lipogenesis. So even though there's no insulin present, they're still somehow activated in a way that they think they should be storing more fuel, even though insulin is not acting anymore. Right? Is that clear? So what I'm trying to say here is that in the normal case, you have a nutrient response that's going to work through insulin, promote lipid storage and glycogen storage. But sometimes something else happens. So you have a different system that can be activated largely in response to obesity, and that can maintain the rates of lipogenesis and glycogenesis independent of what insulin is actually doing. So in the context that I've shown you, insulin signaling is way down, but we're still able to get extant increases in lipogenesis and glycogenesis. So we've reasoned that there must be something else, some sort of insulin-dependent, independent function that could promote lipogenesis and glycogenesis in the obese state chronically, even when insulin's not present. Okay? And it's going to be mTOR. I'm not going to kid you. We're not in a couple of slides, I'm going to tell you that I think it's mTOR. So one of the reasons we think this is if we look at this now in isolated cells, These are mouse embryonic fibroblasts where, again, we deleted TSC2 either in the white bars or in the only bars on the top. And then we measured the steady state levels in these cells of lipid and glycogen. And so you can see there's a big increase in steady state lipid and a big increase in steady state glycogen. So again, these cells have chronically activated mTOR. These cells think they're in a level of nutrient excess, even though they're really not. So they have this big increase in the amount of stored nutrients. And if we look at the rate by which those nutrients are stored, again, by tracer studies, either into triglycerides or into glycogen, the rates of both of these storage events are increased just by activating mTOR. Okay? So mTOR is sufficient to increase lipogenesis and glycogenesis in this cell culture system. So again, what the model that we're thinking about is that overfeeding can activate mTOR. Now I'm talking about in fat, although previously I was talking about in muscle. And we think that that might be one of the reasons why you get this increased lipid storage. We also think this is one of the reasons why when you're obese and you have this chronic elevation of mTOR, you may continue to have lipid storage even when you shouldn't have insulin action or you don't have insulin action. And again, there could be something else, which we haven't ruled out yet, but the paradigm we're thinking again is if we get rid of something else and we start just by activating mTOR in fat, we can ask, well, what happens to lipid storage in an intact animal? I've already shown you this happens in cells, so we want to know what happens in vivo. And again, these animals are going to be on a normal chow diet. There's nothing special about what they're eating. And so what I'm showing you here, again, is some MRI data from some of these animals. And you see they have about a 30% increase in the amount of fat stored. So they're eating the same food, but they're more able to store fat. Okay? So they're more lipogenic in their adipose tissue. So we're a little farther along in this to try to understand exactly how this happens. So what we want to know is something is happening downstream of mTOR, which is somehow promoting lipogenesis. Now, the thing that we think mediates this process is a transcription factor that's called SREBP, so sterile response element binding protein. And it's a known mTOR target, and it's not known how it's actually regulated, but it's known when mTOR is active, SREBP is activated, and that can drive lipogenesis in and of itself. So the reason we thought SREBP could be promising is we look at the levels of SREBP mRNA, and SREBP has a positive feedback loop where its own mRNA responds to itself, or we look at the levels of SREBP protein, we see the same sort of thing I've been telling you about mTOR activity and nutrient storage. So even though SREBP can normally respond to refeeding, if we compare here to here, there's sort of this insulin-driven response to SREBP. When you're in a high-fat diet state, you have this hyperactivation of SREBP at both the protein and the mRNA level. And again, this is kind of consistent with what I've been telling you about mTOR. Even though insulin in the right-hand case, in the black bars, is working less, it's still able in the high-fat diet case, refeeding and mTOR activity correlates with this increase in SREBP. So it's not directly due to insulin, but there's some other signal that is activating SREBP. So we think that signal was going to be mTOR1. So to test this in cells, we repeated that lipogenesis assay that I showed you a couple of slides ago, where if we look at the wild-type cells in black, or in dark gray, and then the TSC knockout cells, when we activate mTOR, they have this increased rate of lipid storage, of lipid synthesis. But if we knock down SREBP1 by shRNA, we don't see this increase in lipid storage at all. So what this suggests to us is that the way that mTOR, at least in these embryonic fibroblasts, can increase lipid synthesis is dependent on SREBP. Okay, so you need SREBP to get this effect. And so we have sort of two outstanding questions about this. One is there was a big question mark between mTOR and SREBP. So how does that exactly happen? So we're working on answering that question. But the second side of that, of how does SREBP regulate lipogenesis, that's pretty well known. It's a transactivator of a bunch of lipogenic genes. We see induction of lipogenic genes. We knock down SREBP. That induction is largely ablated. So there's another way that mTOR can regulate fat storage, and that's by regulating the rates of adipogenesis. So everything I've shown you before is irrelevant of new fat cell formation. That's what we call new fat cell formation is adipogenesis. The reason I'm saying it's independent of that is the way that we engineer our knockout is mTOR is not activated until after they become a mature fat cell. So we're not affecting the rate at which our cells can become adipocytes. We're just affecting cells that are already adipocytes. But independent of that, if we look at cultured 3T3L1 cells, they grow normally like this as sort of fibroblastic cells. If we give them this differentiation media, you see they become fat cells. This white stuff is basically all lipid accumulation. They accumulate a ton of lipid. But if during that process we inhibit mTOR by giving rapamycin, we've completely blocked this differentiation process, and we block the induction of a series of these adipogenic genes. So that's the other angle that we're trying to understand of like how mTOR regulates fat storage, is by understanding how it regulates adipogenesis. Now again, this doesn't affect our TSC mice, it's irrelevant for them, but if you're somebody who's gaining weight, it's sort of relevant for you. So we think mTOR can have sort of two different effects as it relates to fat storage. One is at least through SRNP, the transactivation of lipogenic genes, and then a second is either through SRNP or independent of SRNP, an induction of new fat cell storage. So what we're saying is you get more fat cells, and then those fat cells are able to store more lipid, and those two things combine to give you both more adipocytes and more bigger adipocytes, more fat in those adipocytes. So that's how we think that mTOR, even in the absence of insulin, in the case of obesity, can promote excess fat storage. Maybe in a way that we would not like that to happen, but maybe evolutionarily would have been advantageous in a way that we probably were always trying to come up with ways to store more nutrients, because we were never sure when we were going to find the next saber-toothed tiger or whatever it is that we ate when we were evolving. Okay, so what I've tried to tell you, I don't have a clock here, so I have no idea how I'll do it for time. Oh, it's perfect, 45 minutes. Is two things. So one is that, well, three things. One is that mTOR is a really important nutrient-sensing enzyme, and its role is context-dependent. So the way it works is different in different cells. Now again, this may seem obvious to you. If that's the case, you're all much smarter than I am, but it took me a few years to get here. In muscle, we think the way that mTOR works is to regulate oxidation. So we think it increases energy expenditure. We think it does that by creating more mitochondria in the muscle. And we think that in that context, it plays a role in the adaptive thermogenic response to hypernutrition. So that's what we think it's doing in muscle. So in muscle, it's largely catabolic. Mice end up being skinnier. In fat, we think it does the opposite. In fat, we think it tells the fat cell to make more lipid, make more adipocytes, make more lipid, and therefore is obesogenic instead of promoting leanness. All right, so we started this work when I was a postdoc in Alan's lab. So he paid for the first little bit, and I had another postdoc and an undergrad, a PhD student that was working with me. And then we continued this in my lab with a postdoc and a grad student and a couple of other people that have helped on this project. This was funded by sort of startup funds from Lavonder Children's Hospital and Memphis Research Consortium, and then NIH funds. If you have any questions, I'd be happy to answer them.
Questions?
So you've activated TORC1 with the TSC knockout in adipose and muscle specifically. How do your data, are they similar or different in terms of raptor knockout in muscle or adipose?
So that's a good point. So raptor is an essential component of the mTORC1 complex. So the way that you would do the opposite experiment that I showed was be to get rid of raptor in this tissue-specific way to keep mTOR off. So the raptor fat-specific knockouts match really well. So they're very lean, they don't get very fat, they're resistant to adipogenesis, so they're very skinny, which is kind of the opposite to our mice. So that seems reasonably straightforward. Muscle raptor is a little confusing. So mTOR also plays a role in the new muscle formation. So when you knock out mTOR in muscle, it actually impairs your ability to make new muscle, either to make new muscle or repair damaged muscle. And those animals actually die pretty rapidly. So they all die within, I think, 8 months or something. And they're very sick leading up to it. So they have sort of horrendous muscle atrophy. So they do have lower energy expenditure, which would be kind of consistent with what we're saying. But because of all the confounding problems of muscle development, we don't really know. So the way we're doing it, and we're not there yet, but the way we're actually halfway through doing it is to get the animals to be adults and then knock out raptor using doxycycline-inducible knockout. So we think that'll be cleaner because then we don't have to worry about this developmental myogenic effect. We only need to worry about what happens metabolically after that. But we don't have any data on that yet. But it doesn't kill dox doesn't kill the animals, so we think we could probably get something. We just don't have much yet.
So thanks, I really enjoyed it very much. The increase in torque in muscle, the increase in thermogenesis. What are the ATP requiring processes that are upregulated? Just having more mitochondria,
Is that
you're making the animal more inefficient, correct? So what are the ATP requiring processes that you think are enhanced in that?
Right, so that's a good question, and that's a question we don't have a really great answer to. So one of the things we thought was there would be an increase in the uncoupled mitochondria so that it was going to be basically burning that off as heat. So as you know, UCP2, UCP3 may or may not even be uncoupled to begin with, so we didn't really see any change with that. We've been trying to measure this in isolated muscle strips using a seahorse, That actually turns out it's a little tricky because even tiny little mouse muscles are a little too big for our 96well seahorse apparatus. So the answer is we don't really know. Like, we don't really know what's happening. We know a lot of transporters, amino acid transporters and fatty acid transporters are upregulated. And we know there's more mitochondria present. We don't know much about the efficiency of these mitochondria. We would argue that it's probably really poor in terms of ATP production because you're not just going to generate all this ATP and then not use it for anything. We've looked at AMPK activity. AMPK activity is down, which is sort of consistent with more ATP, but that's also confounded by mTOR has direct effects on AMPK. So I don't know if that's a measure of ATP levels or not. We've been meaning to measure ATP levels. We just haven't gotten to that yet. But I would be, you can't chronically just keep making ATP forever. It has to be used for something. And so our guess is that somehow mitochondria is losing that ability to make ATP, and then it's just being uncoupled as heat somewhere. But since it wasn't UCP, it's exited that realm of the thing we thought it would be to the realm of it could be almost anything else, which we're trying to sort.
Now, this might be a very obvious basic question. What is known about mTOR activation in humans and how different people may have different responses?
Yeah, it's a good question. So obviously, people have different thermogenic responses. That's a thing that people know. People have different responses to diets too. So some people are very responsive to high protein diets. Some people are kind of resistant to high protein diet inductions. We don't have any humans. The place where I'm at in Memphis is a pediatric place, and it's very hard to get biopsies from kids. So we don't actually have any positive data right now about mTOR in humans. What we would like to know is, I guess, two things. We'd like to start from, are the things we're seeing in mice actually true at all in humans? So we've been working with some people at Pennington to try to get some of their muscle biopsies where they've done some of these overfeeding studies to see if that's increased. Now, I guess the more relevant question would be, what if we had a knockout human? And what would that person be like? So unfortunately, if you knocked out Raptor, you would be dead. And if you knocked out TSC, you would also be dead. And so those humans are probably not going to be available to us. The one model that we've been looking at a little bit is there is actually a genetic disease that's associated with the loss of TSC. That's why it's called TSC. It's this disease called tuberous sclerosis. So those people are heterozygous for TSC, and then they randomly pick up a secondary sporadic mutation of their good TSC copy, and then they have all sorts of problems. They get tumors and autism and growth and things like that. We know that if we take cells from their tumors, those tumors are very metabolically active. If we reintroduce TSC, their oxygen consumption rate decreases quite a bit. Now that's not a great answer because it's like a tumor from a random mutation and obviously not a muscle yet. But that's kind of as close as we've gotten. There's associations of Raptor for predispositions to obesity as well, which is kind of consistent with what we have, but not obviously not as clean. And we would actually argue that inhibition of Raptor genetically, where it's going to affect every cell in the body, is going to be kind of impossible to sort out because we're just of the two tissues I've talked about, they have totally different effects on BMI, so it kind of depends on which is the major driver in humans.
Some questions if that over. Go ahead.
So I have a couple questions about your adipose tissue side of the story. Mm-hmm. So perhaps I missed it. You upregulated mTOR in adipose tissue and saw these great effects, but I didn't remember. Does overfeeding actually increase? Did you show data showing
that it increases mTOR activity
in adipose tissue?
Yeah. So I didn't show it, but yes, that's true. So it actually increased chronic high-fat diet feeding increases mTOR activity in all three tissues that we've looked at. We have another whole story I didn't tell you about the liver, which is maybe more confusing, but in all three of those tissues, mTOR signaling is elevated. Now, we've been trying to parse out, what does chronic mTOR activity, say in response to chronic overfeeding, mean relative to acute mTOR activity, which happens whenever you have insulin? That's a little trickier to tease out. Certainly, our model is a chronic model. So our model sort of better matches the idea of chronic overeating. But kind of one of the neat/confusing things about mTOR is even though in our mice, when we have it chronically elevated by deleting TSC, it can still respond to the other signals. It can still be modified by glucose levels and by amino acid levels. So it's not like it's chronically on full blast. It's up, and then it can still kind of go up and down in response to sort of other nutrient cues, which is a little different than, say, AMPK, which doesn't have that kind of fine control.
And then my second question on that side of the story is you refer to creating an obesogenic environment. But to me, I guess my reframing of that is that you're actually just enhancing the capacity to store fat rather than the energetics of fat storage. So it's not making them more obese, right?
Right,
Because
it's
it's a
making them more
question.
able to be
They can store more fat, which may actually even be
beneficial.
favorable.
Right. So, and so Jeff and I talked about this a little bit earlier today. One thing I'm not talking about any of these is that even though some of these mice are really skinny and some of these mice are really fat, from an insulin signaling perspective, they're all effectively insulin normal sensitive. And in fact, the adipose tissue knockouts, which are fatter, actually handle glucose in response to insulin a little bit better. And that's, I think, what Jeff's kind of getting at is this idea that having an excess ability to store excess fat can actually be beneficial to some level. And I think that's what this is, because this is a fat-driven model. Fat's backward, adipocyte-driven model, where we are affecting the adipocytes. Now, granted, if you eat a lipid diet, that's going to have effects on the adipocytes, but then that's also going to have all these other fat effects. Yeah, so that's a good point. And one of the ways that we think TZDs are beneficial is basically by doing that, by increasing the ability to store fat. Even if it makes you a little bit more obese, it makes you better able to handle glucose. But going back to the catabolism point, I made at that one initial slide with things separated. I kind of think that figuring out ways to make better fat storage is like sort of a short-term solution to a long-term problem. Whereas figuring out a way to make people burn that fuel is sort of probably the only way that sort of chronic, stable weight loss is going to be able to be maintained.
Yeah, so go back to your slide where you have the differences in the adipocytes, fat pads. With that, it was in the beginning of the talk.
All right. So, a little recap for everybody here. Probably could have gone back to the beginning.
Here you go.
Yeah.
Okay. So these are huge differences as a consequence of knocking out TSC1 in the muscle, and the fat pads are smaller because there's less triglyceride storage. Okay. So there appears to be less of a difference with your brown adipose tissue shrinks as well. Is there any evidence of this is kind of off topic, beiging of these tissues as a consequence of this?
Yeah, so that's a good question. I would say almost every single time I show that picture, somebody asks me, "Oh, are you beiging your adipocytes?"
Yeah.
So what beiging of adipocytes means is sort of this idea that you can increase mitochondrial biogenesis and uncoupling in fat cells as a way to burn fuel. This has been an attractive theory that people propose as a way to increase your energy expenditure. So if this was a whole body knockout mouse, and we saw this, we might immediately be like, oh yeah, maybe that's what's happening, is that mTOR is regulating beiging in adipose tissue, and therefore that is the driving phenotype. The confounder, there's a couple of confounders with what we have about is it, so first of all, we don't think it's driven by beiging. The only reason we think that is because it's driven by the muscle. And so unless the muscle is causing the beiging, we don't think that it's directly an effect. If this was the case of the adipose tissue knockouts, we would maybe argue that it could be that. The confounding part is pretty much any animal that loses fat mass is going to have beigeur adipocytes, because now you have less fat. So per unit fat, you've got more mitochondria. So if we look at, say, the levels of UCP1 in these animals, they're actually they're up. And if we knew nothing else, we might be like, oh, this is because we've upregulated UCP1. But it's kind of confounded by the fact that now the fat tissue has less fat. Also, the animal is not that well insulated because it's lost all that insulating fat. And that's going to make them a little more thermogenic in their adipose tissue too. So I try not to mention UCP1. I have some doubts about whether beiging of adipose tissue can actually make you lose weight in the first place. But certainly in this model, I definitely have doubts.
And my other question had to do with mTOR activity in subcutaneous versus visceral fat adipose tissue depots. Is there a difference, or
so we find it's a little by high-fat diet. We find it's a little more activated by high-fat diet in the subcutaneous fat than in the visceral fat. It's activated by both. It's just a little more responsive in the subcutaneous fat. We haven't looked at the high-protein diet, which I think that would actually be a bit of a cleaner experiment. Would be whether there's differential effects in both. Yeah, so it's kind of up in both, a little bit more up in sub-Q. Yeah, we're really interested in that idea of differences in the depots. Actually, in both of these models, we're seeing really no differences. And that's another reason we don't think that this is driven by some change in the adipose tissue, because there's some biological depot differences between sub-Q and visceral fat, so that, for example, we have this other project for glucocorticoids where we see a really dramatic effect on subcutaneous fat, almost no effect on epidermal. In this case, we think since it's not driven from the actual fat pad, differences in the fat pads are less relevant because the muscles just make sucking it out both at the same rate.
Any other questions? All right. Oh, wait a minute. Go ahead.
We have one more minute. And then
So your macronutrient, the story you opened up with macronutrient balance, the squishy on that in terms of longer term, out a year, 2 years in terms of the macronutrient balance. And the more recent study that you're probably familiar with Kevin Hall's very controlled conditions of changing fat, they kept protein the same, but changing fat content in the diet did not affect energetics, the VO2 was the
loss of
same.
weight. Right.
And the amount of weight loss was identical, or even the high carbohydrate or reduced fat diet actually made them more energetically favorable. So that seems to be contrary to.
Well, I wouldn't say it's contrary because, I think if we were just talking about the high fat diet versus high carb diet, it would be contrary to that. And in his case, so he's talking about this is a paper in Cell Metabolism, I think in August. And it was a very, very well-controlled weight loss study where they made them lose weight on controlled different diets. Now, I think the important part is he didn't change protein. My guess would be if he changed protein, he would have seen the things that we saw. And I've talked to him a little about this, and he said, yeah, that's one thing that we'd look at changing. So his takeaway was that because fat, at equal lowering of carbohydrates, it didn't matter if you took those carbohydrates, equal lowering of calories. It didn't matter if you took them from carbs or from fat. You got sort of effectively the same effect, maybe a little better with fat. Now that, I don't think anything that I maybe have proposed is necessarily contrary to that. He just didn't do that third arm. The other caveat to his thing is his diet was really short. It was very well done, very controlled, but it was like a week. And so whether that is the same as like a long-term diet, I think there's probably some sort of glycogen-dependent changes that are associated with its low carb thing. But it's a big question. Is a calorie a calorie? Like it seems kind of astonishing that at this point in metabolic research, there's like still maybe, I don't know, kind of. And that's a pretty fundamental question of are these things actually handled in the same way?
All right.
Okay, thank you very much.